Okay, here we are. I have the pleasure to introduce our first presentation, Dr. Amutha Boominathan. She joined the organization in 2013. She's the group lead for the MitoSense program and has over 22 years of experience in mitochondria biology. She'll be presenting on allotopic expression of mtDNA genes. You can also read her presentation summary and bio on the event's website. I want to say we're going to have a Q&A after, and please make sure to submit your questions in the chat called Main Stage. So if you go to the chat icon on the left-hand side of the screen, click on it, and then the subgroup Main Stage. So let's welcome her to our stage right now and enjoy the presentation.
Hello, everyone. A very good morning. I hope you enjoyed our yesterday's session. And today we are hoping to hear some more very exciting presentations. As Maria introduction, I have been leading the MitoSense project at Sense Research Foundation. This is one of the longest running in-house projects initiated by Aubrey de Grey, and I am happy to share our progress, challenges, our current strategies, and how we would like to expand this project. As you all know, the Sense's view of mitigating drivers of aging is by addressing how we can remove the damages that occur in our cells as we age. We have different strands along this, and I will be talking about how mitochondrial mutations that accumulate with time contribute to the aging process and how we can overcome them. The title on this slide is probably, it's out of a textbook from high school, that mitochondria are the powerhouses of the cell. But what you will appreciate is that mitochondria are the hub for many, many more metabolic pathways than just ATP production. They are the center for calcium and hormone homeostasis. They are the site for iron sulfur cluster production and lipid metabolism in the cell. But what we also need to appreciate is that in the eukaryotic cells, in the mammalian eukaryotic cells, this organelle comes with its own DNA. This is a small circular double-stranded DNA of about 16.5 kb in humans. And this feature is very important for its function. Just as other power plants, regular power plants, these little power plants in our cell also produce byproducts of the energy generating system, and these byproducts can result in deleterious events for the DNA in particular. And this double-stranded circular DNA encodes 13 very important protein coding genes. They all assemble into the respiratory chain complexes that's very crucial for energy generation. But in addition to just this accumulating mutations in the mitochondrial genome, the mitochondrial function also is known to decline with age. So here is a little statistic on the disease population. Now, the first point is genetic disorders. So 500,000 people are diagnosed with mitochondrial DNA disease worldwide. That is mostly uh, a pediatric population that's born with the mutation. But it is estimated that it is likely that 1 in 200 births are born with a mitochondrial DNA effect, where the phenotypes may be much milder, but it's there in the population. But a general decline in mitochondrial function has been attributed to several neurodegenerative diseases, and you will appreciate the scale of this effect. So there's approximately 7 to 10 million people estimated to have Parkinson's disease, and almost 55 million people have dementia-related issues as we age. Here is to give a context of all that. On the left, I am showing you a general decline in function with aging. We all know about sarcopenia, but what has been observed in sarcopenia is that, let me get my pointer here, that the mitochondrial content composition is altered when from a young muscle to an old muscle, and there is a connection between the neuromuscular connection is very much related. So there is even altered mitochondrial function with respect to size and their ability to perform biochemical reactions in the nervous system and in the muscular system. On the right, I show you a typical mitochondrial DNA disease, and this is resulting from a mutation in the mitochondrial DNA, and it results in muscle myopathy. And the bottom figure is control tissues stained for, COX, the complex 4 and succinate dehydrogenase enzymes, which is part of the respiratory chain, and you should get these distinct brown colored staining. But in the disease muscles, you can see that it's much altered with these blue punctate structures in between the cells. To give a little more context, I'm listing only some of the diseases associated with particular mtDNA mutations observed in patients. There are rearrangement mutations where, a small portion of the mitochondrial DNA is either deleted or rearranged, and this results in diseases like progressive external ophthalmoplegia, Kern-Sire syndrome, which is a classic deletion of approximately 5 kb of the mitochondrial DNA, Pearson syndrome. And then you can have mutations, particularly point mutations in the tRNAs or the rRNAs of the mitochondrial DNA. Now, point mutations in tRNAs will globally affect the protein translation within the mitochondrial matrix, general decline, general disruption in the translation process, and several pathological conditions have been reported, such as MELAS and MRF. Now, there can be point mutations in the 13 protein coding genes, and these also result phenotypically. They can have the same conditions as MELAS or MRF, but they also result in those are the phenotypic display of the mutations. Why I highlight these is that we use some of these mutant forms to test our gene therapy approach to it, which I'll come in a little bit. Again, when I told that mitochondrial dysfunction function can be is also the cause for larger diseases like Parkinson's and Alzheimer's. In addition to the disruption in mitochondrial biogenesis or the mitophagy that's required for turning over mitochondria, there are certain mutations that has been observed in patients with Parkinson's or even Alzheimer's. To highlight, even pink and parkin are mitochondrial proteins. I also want to point out here that in the mammalian cell, the half-life of mitochondria is estimated to be around 10 to 14 days, in that they are recycled every 2 weeks or so. So when there is a disruption in this turning over, then you can imagine what cascade events that can have. At Sense Research Foundation, we are addressing this from 3 different angles, and there are many, many other angles other researchers are exploring. But our primary project has been to develop gene therapy for mtDNA myopathies. Our goal is to find optimal parameters in expressing these 13 protein coding genes externally, not from the mitochondrial genome, within the cell, and achieve function. We have recently started 2 smaller projects, the Gene Drive project and mitophagy, which I don't have time to elaborate for this talk, and it is also less developed, so I hope I will have an update next time, maybe in a year or so. But as I mentioned, there are alternative methods that other researchers are exploring. This is how we can augment general mitochondrial biogenesis, external organelle replacement, which has taken off in a big way. You might have heard about this as mitochondria replacement therapy. Several startup companies have started in the past few years, including Minovia, Selvi, and Mitrix. There are compounds. Of course, exercise is known to boost mitochondrial biogenesis, so people have tested that even the clinical trials. And exercise mimetics, small molecules to mimic that, and antioxidant supplements. So how do we achieve a gene therapy for mtDNA myopathies? We will address this as allotopic expression, that is placing the wild-type version of a mitochondrial gene in the nuclear cytosolic compartment and its efficient targeting to mitochondria. Why would we place it in the nuclear cytosolic compartment? So there are some key differences between the nuclear genome and mitochondrial genome. As I spoke earlier, the mitochondrial genome is a naked double-stranded circular DNA. But because of its prokaryotic origins, it also has much less repair mechanisms or protective proteins when it is not replicating. But one more important thing to remember is that for the nuclear gene, it replicates only once the cell divides. However, the mitochondrial DNA divides many more times. It doesn't synchronize with the nuclear DNA replication, and it is observed that these mitochondrial DNA accumulate 10 times more mutations than the nuclear genome. That's primarily because of errors in the replication and inefficient repair mechanisms to repair those errors as we progress. And of course, one major issue is proximity to ROS, or the byproducts of the energy generating system. The DNA is very close to the sites of ROS production. And if we were to edit the mitochondrial DNA, that's a very challenging task that you would have to, even for one single cell, you would have to have thousands of those editing events to happen for functional outcome. People have tried CRISPR, but CRISPR does not work yet, although certain base editing enzymes have been identified recently, and but then they work only for certain mutation types, and then the efficiencies are still very poor. People have tried other DNA editing approaches like mitochondrial restriction enzymes that if the resulting mutation results in introducing a unique restriction site, we could target that restriction site, append it with an MTS, and send it into the mitochondrial matrix, and thereby eliminate mutant mitochondrial DNA. But this is counterproductive in itself, that if you have a very high mutation load, then there is not enough good copies of the mitochondrial DNA to take over and bring back function. So with all these caveats, the best way to approach would be to place um, these mitochondrial DNA protein coding genes in the nucleus, find the appropriate conditions to express them from the nucleus, and try to retarget the proteins to the organelle. Now, evolutionarily, mitochondria have achieved this. The proteome of mitochondria is, it has more than 1,400 proteins in it, of which, except for these 13 proteins, all the other genes are nuclear, originate in the nuclear genome, and then they have very specific targeting parameters, and then they assemble into the organelle. What are the steps to successful allotopic expression? First of all, we would have to read nuclear language, to in a little bit an appropriate construct that you did with the appropriate with the correct targeting elements, and that's recoded for the nuclear context, and you can stably integrate, or you can express episomally, that's fine. But then you would want the resulting mRNA to exit the nucleus. The export should be efficient, and translation happens in the cytosolic milieu, because they have now that we have appended a mitochondrial targeting sequence. This targets it to the mitochondria, and the protein should go and assemble into the four respective oxphos complexes to achieve function. Just to highlight that we published a review article on the current literature on mitochondrial DNA mutations, their pathologies, and how we can address them recently in biomedicines. So now for the actual steps here. What do we mean by realigning the mitochondrial and nuclear languages? As I mentioned earlier, this is the structure of the human mitochondrial genome, and all the genes, the protein coding genes, and the tRNA but there are some um, fundamental differences in the codon usage between the mitochondrial DNA and the nuclear DNA. So if we were to place the protein coding genes for mitochondrial DNA as is in the nuclear context, there will not be productive translation. For example, in the mitochondrial DNA, AGA and AGG are terminating codons, whereas in the nuclear context, they code for arginine. And ATA is methionine, and often even we start methionine for some proteins in the mitochondrial context, whereas it codes for isoleucine in the nuclear context. And tryptophan, the TGA codon, mitochondria actually have two codons for tryptophan, and TGA is actually a terminating codon in the nuclear context. So for to begin with, we would have to realign at least these parameters, these codons, for productive translation in the nucleus. But we observed that just recoding the mitochondrial DNA is not as efficient. So what we did is we did something called codon optimization. Now, codon optimization is not necessarily a very, I mean, it has been implemented previously in the industry to produce protein therapeutics, like if you wanted to achieve efficient, large scale production of proteins. But what this does is that it and several companies offer this codon optimization protocol, and sometimes what the algorithms that they use are proprietary, so we are not exactly sure what all features have been integrated, but we did this using these are some of the features that JunScript takes into account when you doing codon optimization. It optimizes for GC content, mRNA secondary structure, removes cryptic splicing sites, premature poly A introduction sites, and so on and so forth. I'm just going to explain using one of the genes what codon optimization really does in our context for the mtDNA genes. This is the ND1 protein, and it's about 318 amino acids. And if we take, for example, valine, there are 4 codons that can code for valine, and in the recoded context, this last codon is practically absent. Here is the scale for the observation of these relative codons in the gene. Codon optimization, when it does codon optimization, practically that codon is represented much more significantly than that in the recoded version. Of course, there is a combination of all of this for several of the amino acids, and we used this um, algorithm to for as an initial step to achieve protein expression. That's the first part of it, and then obviously we need specific import signals to tell that these proteins are actually going to go to the mitochondria. Um, Now you can so this is our base construct. We have an appropriate promoter. This is actually a viral promoter that I'm talking about here. We have the CMV promoter. We have a mitochondrial targeting sequence. We used in this case the MTS from ATP5G1. This is a subunit of complex 5, and in literature it has already been reported that it works. So we went with that mitochondrial targeting sequence, and we synthesized the recoded or the codon optimized versions for all the 13 genes. So we constructed 26 different plasmids, and to track them, we introduced a flag tag at the C terminus. Um, what you can appreciate here is that each protein, all these proteins integrate into the inner membrane of the mitochondria, and they are all membrane proteins that and spanning the inner membrane, you know, one for small proteins like ATP8 to several transmembrane domains in some of the longer proteins, and they come with their own physical properties for the protein as well. So you can imagine our tall order to develop gene therapies for all these 13 genes, but our immediate goal is, if we can find the optimal parameters for each and every one of them, we may be able to help at least the pediatric population that come with very specific mutations in any of these genes. So what happens at the expression level? We published, this was the work of and Bhavna Dixit. The blot might look a little busy, but what I want to draw your attention is that in every case, the codon optimized construct produced appreciable protein quantities, and it associates with mitochondria. I'm going to be careful in the word associates, because we know that of these 13 proteins, even though all 13 of them are expressed, only 8 of them produce stable expression, and there are certain instances like COX-2, although it produces a lot of protein and it associates with mitochondria, we know that there is a processing problem there, and we hope to address this issue, which I will talk about a little later. Okay, so now we can produce this. We chose one of the proteins, ATP8, to show if this allotopic expression can really rescue function. Towards this, we used a patient-derived cell line, and hybrids are the model systems of choice in mitochondria field, specifically for mtDNA mutations. How these hybrids are generated is that you would fuse patient primary cells with a cell line called row 0 cell line, where there are mitochondria in them, but they don't have mitochondrial DNA. And they also have a nuclear selection marker. So when you fuse these two cell lines, the patient's cytosolic component fuses with the cytosolic component in the row 0 cell line, and it's the patient's mitochondrial DNA that takes over the system, and now you have in the lab we use an immortalized cell line called the 143B row 0 cell line. So now that gives us a chance to maintain them for longer periods of time in the lab and test our constructs. So now, what is this primary cell line? This cell line was originally reported by the Rodenburg lab in Netherlands, and this is a 16-year-old male with a pical hypertrophic cardiomyopathy and neuropathy. They suspected for mitochondrial disorder, and upon sequencing the mtDNA in this patient, they observed that there was a particular mutation at 8529 G2A conversion. Now this results in a truncation of the protein after 55 amino acids. Now the primary cell lines had over 90% homoplasmy. So here is another feature that we have to remember is that mitochondrial DNA, because of the multiple copies that they have, there is something called a heteroplasmy condition. And when the mutation threshold goes over a certain threshold, that's when phenotypes start to appear. So in this case, the mutation load was over 90%, and the resulting in a severely reduced complex 5 activity. So we got these cell lines, and in the mitochondrial genome context, ATP8 and ATP6 are, it's a bicistronic region, in the sense that the mRNA for ATP8 and ATP6 is single, whereas there is an internal initiation site for ATP6 to produce two different products. So in this cell line, we saw that the patient, there was absolutely no ATP8 protein, but ATP6 was also affected because this mutation particularly affects the ATG. It converts that G to an A. So we hypothesized that both, if we were to achieve function, we would have to co-express ATP8 and ATP6. So we did that in these cell lines. What I'm showing you here is a blue native page, that is where you preserve the complexes for the protein complexes in the electrophoretic system. And in the patient cells, even a marker protein for ATP5H, which is a complex 5 protein, is completely absent. So the complex 5 is not assembled properly at all. And when we express ATP8 alone or ATP8 plus ATP6, the complex comes back. This is the wild type cells control. And this is for our flag tag for ATP8 and ATP6. In fact, you can see even some of the dimers. So complex 5 exists in monomers and dimers, so even some of the dimers coming up. So we went, okay, sorry, that's too quick. Yeah. So then we did an in-gel activity assay. Okay, now the protein is present and it integrates into complex 5. Does it bring back function? This is an in-gel ATPase activity. As you can see, the delta ATP8 is the patient cell line. The ATPase activity is in the soluble fraction, or the fraction that faces the mitochondrial matrix, or the F1 subunit. And this mutation, what we are talking about, is in the FO subunit. What you can see is the ATPase activity is there, but it is in a location much below the monomers or the dimers. It's degraded. But once you put ATP8 or ATP8 plus ATP6, number one, the activity comes back in the monomers and dimers. Also, some of the unassembled complexes goes down. The intensity of the unassembled complexes go down. So we were confident that it is bringing back function. We tested this the classical assay for oxygen consumption. That's what we do to assess mitochondrial function. These mutant cells with very low activity, the control cells, 143B cells with wild type mitochondrial DNA is here, and the rescue cells bring back the function nicely. Similarly, we have another assay. It's called the cell viability for OXPHOS restrictive media. We use galactose as the media here. The patient cells die rapidly in this media, whereas the rescue cells are viable. Okay, now we have shown that allotopic expression for ATP8 at least works in vitro in patient hybrids. Can we prove this in vivo? That was our next question. But I have to point out here that model systems for mtDNA mutations are very hard to find. Fortunately, there is one model available for the ATP8 mutation. I want to point out here that all the following in vivo studies were done in collaboration with the Buck Institute, with Dr. Martin Brand and Dr. Birgit Schilling. And David, Bhavna, and Carly were the main contributors to this work. What is this mouse model? This mouse model has a specific mutation in the ATP8 gene. There is a conversion of the fifth amino acid and aspartic acid to tyrosine on the N terminus of the protein. This model was reported by the Ibrahim group from Germany, but so there are some caveats to this model. When the Ibrahim group reported this model, they reported a very significant behavior phenotype, anxiety phenotype in these mouse models. But in our hands, we were not able to replicate that behavior phenotype. But we took advantage of the amino acid change here to test how well our allotopic construct can compete with this, and then that way we can show which is likely the scenario in patients as well. Null mutations often don't survive. In fact, the viability for null mutations is very, very less. So there are several substitution mutations or amino acid substitution mutations that impair functions mildly, and patients suffer as a consequence. So we said, okay, yes, we don't have a black and white functional assay, but we can still use this model to test our hypothesis if our allotopic constructs can compete with even existing mutant proteins in the cell. Now, a few characteristics about this mouse model. What they did is, this is the FVB mouse they started with. The FVB mouse has mutations in the nuclear genome, but it also has 3-point mutations in the mitochondrial genome. The other 2-point mutations, other than the ATP8 gene, are silent, and they do not have any influence on the product. One of them is in a tRNA sequence, and the other one is in the ND3 gene. It does not change the amino acid composition of the gene. But what they did is they backcrossed this FVB mouse model with the C57BL6J mice 12 times to kind of eliminate all the nuclear background influence. So now these are called conplastic mice. Now the phenotype is solely from the mitochondrial DNA. Now, since mitochondria are maternally inherited, they took advantage of that and did this crossings over and over again. You have the female FVBs crossed with male C57BL6J mice. That's the model that we are going to use here. And we also did this using the safe harbor approach, in that we placed a single copy of the transgene in the ROSA26 safe harbor locus. Now, that to account for copy number, stable expression, and also mitigating potential insertion site side effects. So how did we do this? Here we introduced the plasmid with the transgene in embryos containing the landing site for the HROSA26 locus, okay, maybe the next figure might explain it a little better. Yeah, fertilized eggs are injected with allotopic ATP8 transgene, and now this is done in the maximally modifiable mouse, which has the insertion sites at the HROSA26 locus. So first we generate a wild type mouse with one single copy of the ATP8 gene in the HROSA26 locus. And then now we use the mitochondrial inheritance patterns to create our transgenic mice. So if you cross males containing the transgenic copy with ATP8 mutant females, your progeny will have a nuclear genome with a single copy of the ATP8, and all the progeny will acquire the mtDNA mutation. Of course, the percentages are only 25% of the progeny gets this, and of course we verified this by genotyping and so on and so forth. I want to point out here that this mighty mouse was developed by Applied Stem Cell, and Sense was instrumental in funding this project. So, yes. How did we ascertain that there is a single copy of the gene incorporated within the HROSA 26 locus? We, of course, we did genotyping through PCR, inside outside primers, part spanning the gene of interest and the HROSA 26 locus itself. You can see the red is the transgenic mice and blue is control non-transgenic mice, and we observe very specific products. We also confirmed that through next-gen sequencing that this is a single copy, and it goes into a specific location in chromosome 6 in mouse nuclear DNA. Subsequently, here is what is the protein expression in this context. So we generated both. Of course, we had to generate the wild type transgenic, and we also had the mutant transgenic. So I'm showing you here data for protein expression from 12-week animals. Only one animal is shown here, but we have done 3 males and 3 females, and we've done this 2 rounds, and I will share some data towards that later. So all the tissues tested showed appreciable expression levels. This is, again, the ATP8 or the optimized ATP8 has a flag tag at the C terminus, and they are expressed in almost all the tissues tested. For time's sake, I'm not going to go over all the other functional assays that we did. We were able to show that the transgenic protein product integrates robustly into OXPHOS complex 5, just as we observed in the patient hybrid cell lines. It does not impair function adversely. We've done several biochemical and behavioral assays. We did the ATPase assay, again, oxygen consumption, metabolic cage assessment, several behavior phenotypes, and we also did a cytokine array to determine how this transgene influences, the immune profile, and we did not see any. Of course, this is a small array, but within that, we did not observe any drastic effects. Okay. Now, what we also, when I told you that that was what I showed you was for 12 weeks. We have tested expression for up to 56 weeks, and we also know that it does not change, like introduction of this transgene does not change fertility. The number of progeny were equal between the wild type and the transgenic mouse, and the transgene is faithfully transmitted up to 4 generations. Now, what we are now the question is, what is the ratio of the exogenous and endogenous protein? I want to say this data we are still waiting from our collaborators. We have the mass spec assay developed. We are just and I can and it is promising, but we are just waiting for replicates to confirm the data. And we hope to, once this data is there, we hope to publish this very soon. So I have taken you through one gene that we were successful achieving gene therapy from in vitro models to in vivo models for the ATP8 gene. Now I'm going to switch gears and show some of the other genes that we are working in the lab. One more gene that is working in our hands is is the ND4 gene. I want to point here that there is currently a gene therapy available for the ND4 gene. This is through Gencyte. It's called Lumivoq. Um, um, and and this addresses one of the uh, patient phenotypes, like Leber's hereditary optic neuropathy affects males, particularly over the age of 40, if they have this mutation in the ND4 gene. Of course, other genes can also contribute to this long phenotype, ND1 and ND6 as well, but ND4 contributes to over 50% of the patients. So what I show here is this is the wild type control line. The red and pink is the mutant line, and the green is the rescue line. So what happens in this mutant lines is that it's a some, because this is not a null mutation. There's a different assay that we have to use to prove that the protein is functional. The ND4 protein in this, or the complex one in these patients, lose the rotenone sensitivity. So what we can see in wild type cells, if you block glycolysis and you add rotenone, it goes down to 18%, and you see the same observation in the rescue lines. Of course, we are reconfirming this data, and we are excited about this result. And Carly is the one. She's pursuing these studies. She actually joined as a postbac fellow, and now is an RA in the group, and you can see the progress she has made. Okay, so what I told you, there are 13 genes that we have to develop gene therapies for. Some of them are working well. Some of them we see protein products. Some of them are very refractory to our technology. So now there are several factors that we are pursuing. Something is role of hydrophobicity. All these 13 proteins are, like I told you, they're all inner membrane integrating proteins with multiple transmembrane domains, and they are highly hydrophobic. And it has been hypothesized that this hydrophobicity prevents them or makes it very difficult to import. So, and of course, several other features, like how can we improve mRNA stability and how can we improve translation. And we implemented something called codon optimization, but is that really the real trick? And I'll come to that in a little bit. And improving the targeting sequences, regulating upstream and downstream features, and excuse me, and improving stable expression as well. So since this is the it's also the student symposia, I wanted to highlight this year's summer student Kiefer Lee. He explored the hydrophobicity factor. What you will appreciate here is that this is a hydrophobicity plot for all the inner membrane proteins in the mitochondria. These are all by the nuclear genome. The orange dots, this by the mitochondrial genome. You can see that all these 13 proteins cluster on the top right-hand corner, indicating that they are highly hydrophobic, except for ATP8. And we were able to achieve very good expression for this, which falls somewhat to the left. But now, once you remove on the right-hand side is a plot with the MTS removed for all the inner membrane proteins, you see that the hydrophobicity increases even more. So we are using computational approaches and wet lab techniques as well to see if we can bring down the hydrophobicity for some of the less performing proteins and see if it can improve import, specifically in the context of COX-2. And without giving away too much, please visit Kiefer Lee's poster. And he tested this hypothesis, and he's done some very good work. And we will be continuing this work in the lab. And please visit his poster. Now, for improving, another highlight is we had two summer students this year in the MITO group, Marek Pinto. Marek had completely computational aspect. What we were trying to do is to improve the mRNA stability and also address this codon optimization parameter. What you see here is that this is again for COX-2. These gray bars indicate the transmembrane regions in the protein, and the peaks that you see in red or blue is the relative abundance of optimal codons. So the red ones indicate non-optimal codons, and the blue ones indicate more optimal codons. So now this is for the recoded COX-2. What you can see here is the codon-optimized version of GenScript is completely optimized all across. And the picture on the right can better depict this. Like, the red lines indicate relatively rare codons in the gene. In the context, it's 228 codons, particularly. Yeah.
Can you hear me? Hey, I just wanted to let you know that you have about 9 minutes, and if you can choose if you want to have a Q&A after, or you want to finish the presentation, that's fine, and then people can find you in the room.
I will be done in 1 minute. Yes.
Okay. No problem.
Yeah. So you can, coming back, you can see the stark difference in these codon usage patterns. So please visit Marek's poster too, and what he has done for the summer. This is to highlight some of the publications from the group. And the last slide, this is our team. And many thanks to our funders, of course, Sense Foundation, Forever Healthy, Foster Foundation, Lifespan.io, and Longicity. And we are hiring. So if you know someone who's interested to join our group, please send them our way. Thank you.
All right. That was amazing, Amutha. Thank you so much for the hard work that you've been doing for so many years for us. We really, really appreciate you as a scientist and as a person. We have a few minutes for a Q&A. Amutha, can you see your chat? The chat.
I cannot see the chat. But Linnea said she would help me with it. But go ahead. You can read if you want. Yes.
Yes. So Phil is asking. He's basically saying that he heard this from Aubrey in 2000 without accounting for the discovery since then that ROS are used as regulatory signals, very likely to upregulate antioxidant defenses.
Yes.
And then he says, what do we know about the mechanism of ROS signaling and about how it will be disrupted by moving mitochondrial genes into the nucleus?
Oh, okay. So I think we will be removing that aspect for the DNA factor in ROS. There are important functions for ROS. Yes, since 2000, several scientific reports have come, but you have to appreciate that if we are able to send these proteins back to the position where they are supposed to go, then it doesn't matter. I mean, the ROS signaling will not be affected. It will still be there, but it shouldn't be affected.
Thank you. We have a question from Caitlin. She says, which region is H17 measuring for each?
Okay, so the H17, so this is again a predictive analysis. People have, there may be several methods to do this, but this is developed by Claros et al. This is local hydrophobicity versus mesohydrophobicity. It's and so it's a local 17 amino acid residues in the context of the entire protein, and it gives a window over 69 amino acids. Uh, like I said, we are using the Claros model to depict the H17 versus mesohydrophobicity.
Thank you. Eric is asking, what was the review mentioned at the beginning again?
Okay, yeah. So we that review addresses it is mitochondrial genome in health and maintenance, and how what is the current status of the different strategies that the scientific field is using to treat this, and where we are in this. That's the review.
Okay. Kaylin says, so average of all 17 residue windows or first TMA?
Can you repeat
coming
that,
back
please?
to her, coming back to her first question of H17 measuring for each. So then after your answer, she says, so average of all 17 residue windows, question mark, or first TMA?
No, it's a sliding window of H17.
17 windows, okay.
Mm-hmm.
Eric says, I meant, what was the name of the review? He was asking before, what was the name of the review you mentioned at the very beginning?
Oh, I can send a copy to him. Name of the review? No, it is published in Biomolecules, and it is from our group. And I'll be happy to send a copy of the review.
Eric, you can email me at outreach@sense.org, and I will facilitate that. Okay, if you don't have my personal email, which will be too long now to spell. We have something else. Let's see. So you have to distinguish between the regulator role of ROS in signaling and the stochastic fumbling of electrons in the ETS during OXPHOS.
Yes.
Also, and in any case, all allotropic expression, is probably allotopic, right? Will do is restore the function of the mitochondria to what they did before suffering mutations. They will functionally be normal, useful mitochondria. Anything to add to that, Amutha?
Yeah, I agree to that. Yes. The other thing is, yes, that's our goal too. Like if we can rescue the function, in the mutation context, if we can rescue the function. But I think this will also give us insights how ROS will be affected. I think that's a very hypothetical question, and my answer is that ROS's signaling effects should not be impaired, but we would have to test that to see. Yeah.
Right. Well, since we have 2 minutes for me to introduce the next speaker, we'll have to stop the Q&A right now. But everyone with questions, please feel free to find Amutha in the room. She will probably be around her research booth in the back of the room, so you can just go to her and ask any questions. Is that okay, Amutha?
Oh, absolutely.
Yeah. Okay, you'll be around the booth, or you can find her in the chat, and you can click call, and you can just connect with her, even if you don't actually know where her avatar is. Thank you so much. Thank you, Amutha, again.